Sorry, I have. Yes, plus in camera. No, okay, thank you. Yes, I know that you hear. Sorry, because I didn't share my screen. I forget to share my screen. Okay, so I will do now.
Assalamu alaikum.
It's okay for now? Okay.
Thanks to the committee of the first International Conference on Environmental and Biotechnology and Biodiversity for accepting my participation to the seminar. My work entitled Algerian PGPA Producing ICEBB2021 confers maize root tolerance to salt and aluminum toxicity. For this, let's start with an introduction. The increase of the world's population and the decrease of arable land due to the climate changes are causing growing concerns over soil sustainability. Among abiotic stresses, aluminum toxicity as a result of the acidification of the soil, industrial pollution, and crop intensification is among the most prevalent constraints. Salinity is another major limiting factor to plant growth and crop productivity. In saline soils, sodium chloride is the predominant salt species, and its effects can be observed by decreased productivity or even by death of the plant. Endogenous ethylene concentration increase by aluminum and salt stress via elevated level of the 1-aminocyclopropane 1-carboxylic acid, the ACC, an immediate precursor of the ethylene. So any control of increased production of ethylene can improve the growth of plants and mitigate the effect of abiotic stresses. The application of the plant growth rhizobacteria, promoting rhizobacteria, is considered as a useful tool to reverse such environmental stressors. It has been well documented that some rhizobacteria contain an enzyme, which is the ACC deaminase, that hydrolyzes ACC into ammonia and alpha ketobutyrate. The aim of this study is to assess the capacity of the strain Pseudomonas plecoxyceti pp20 to alleviate damages on maize root caused by salt and aluminum toxicity. The first part of the work was to assess the tolerance of the strain pp20 to high concentration of sodium chloride. For this, bacteria were grown on king bee medium containing different concentration of sodium chloride for 24 hours and at 30 degrees Celsius and at rotation of 120 rounds per minute. As you can see here, the strain PP20 was highly tolerant to sodium chloride, and its tolerance was attained the 600 millimole of NaCl. Same, we assessed the tolerance of the strain to sodium chloride now under the same condition of culture medium, but with a difference of the solution. Here we use the sodium chloride with a concentration from 0 to 5 micromolar sodium chloride. can see also that the strain was highly tolerant to sodium chloride, and it could even grow under a concentration of 500 micromole of sodium chloride. Then we assessed the capacity of the strain PP20 to degrade the ACC in the cell pellet and in the cultured medium. For this, bacteria were grown on king bee medium in the presence of the ACC, and after 24 hours of incubation at 13 degrees Celsius and at 120 rounds per minute, we evaluate the degradation of the ACC by the bacteria. And here we used two concentrations of the ACC, 0.5 millimole and 1 millimole. And as you can see here, the degradation was very, very important in the pellet. It were in the order of almost 5,000 nanogram and 6,700 nanogram per pellet. However, this capacity of degrading the ACC was very, very low in the culture medium. Then we evaluated the influence of the same strain on the root development. And for these, seedlings were grown in filter paper for 5 days after sowing under a condition of sodium chloride with a concentration of 1,150 millimole, all under a condition of aluminum toxicity in presence of 19 micromole of sodium chloride, or even under control or under normal condition. And here, as you can see, the application of the strain PP20 improved the growth of the roots, of maize root, under the control condition without stressors and under salt condition and also under aluminum condition. After that, we measured the root growth parameter. For this, the root length, number and length of the seminal root, and the number of lateral roots, in addition of the root dry mass, were evaluated. And as you can see here, each application of the bacteria PP20 will give a significant difference and a significant improvement for the root. All the parameters were assessed by the t-test, and as you can see here, almost all the measures were significantly different due to the application of the strain PP20. As a conclusion, we could say that the application of a new rhizobacterial strain isolated from Algeria, Sahara PP20 allowed us to evaluate phytostimulation activities and stress mitigation of this strain. A significant difference was recorded between plants treated with bacteria and their respective controls. In the present world, we have reversed the negative influence of salinity and aluminum toxicity root development on the maize plants. We have demonstrated that inoculation with the PGPL PP20 protects plants against the inhibitory effects of sodium chloride and aluminum chloride on maize root growth. The salt and aluminum tolerant trait of PP20 may also contribute to the observed phenotypes. And thank you for your attention.
Thank you. Thank you, Mr. Azdin Zarruq. So, there any question?
Yes.
Mr. Saadullah Mohammed.
Salam alaikum, bonjour tout le monde. Donc, je dois féliciter quand même le candidat pour ce travail qui vient. Je voudrais demander pourquoi utiliser des rhizobia ou des bactéries rhizobium dans le cas des céréales. Le maïs est une céréale. Est-ce qu'il a vérifié si ces bactéries intègrent les racines? Parce que dans la lecture, j'ai vu que des fois, il y a des bactéries qui s'introduisent, des bactéries rhizobia qui s'introduisent dans les racines et qui améliorent un petit peu le rendement en azote. Ça, c'est ma première question. Ma deuxième question que je voulais demander, 800 millimolaires, est-ce qu'il ne trouve pas que c'est une concentration, est-ce que vous ne trouvez pas que c'est une concentration un peu élevée pour la tester sur une croissance cellulaire? Parce que 800 millimolaires, je pense que c'est assez élevé comme un ACL, concentration d'un ACL assez élevée, qui inhibe en vérité, qui est un peu délétère pour la croissance cellulaire. Merci.
Azdin.
Salam alaikum.
Salam.
Donc, c'est juste pour confirmer. Arabe, français, anglais ou bien comme je veux?
Comme vous voulez.
Oui, c'est par
Ok.
rapport à l'auditeur qui a posé la question.
Ma'alish. Donc, merci, M. Saadallah. Donc, la première des choses, c'est que les bactéries promotrices de la croissance peuvent intégrer les cellules racinaires comme elles peuvent agir juste en surface. Donc, ça, c'est la prochaine étape, Inch'Allah, que nous allons évaluer. Mais pour évaluer la capacité de promouvoir la croissance des racines, donc, ça a été testé au niveau des racines, donc en conditions sous serre comme en conditions de laboratoire juste pour 5 ou 7 jours après la germination, comme nous l'avons également testé durant tout le cycle de croissance du maïs. Et pour tout ça, la bactérie a été promotrice de la croissance du maïs. Donc ça, c'est pour la première question. Pour la deuxième question, nous avons mis plusieurs doses de l'NaCl pour étudier la résistance de la souche bactérienne. Et effectivement, 800 ou bien même 600 millimoles de NaCl est une concentration très élevée. Mais comme nous avons trouvé des résistances de la bactérie avec des concentrations moins élevées que cela, alors, nous avons dit pourquoi ne pas tester à des concentrations de 500 et 600 et même à 800 millimoles de NaCl. Donc, la résistance a été arrêtée, la bactérie a été résistante à 600 millimoles, pas à 800 millimoles. 800 millimoles, c'était la souche de référence, l'Escherichia coli. Et merci.
Merci, monsieur.
Okay. So another question from Mrs. Nafila Zouaghi. Please.
Merci, je vous remercie. Moi, je vous remercie pour votre travail, il est très intéressant. Mais j'ai une question, comme je suis une chimiste. J'aimerais bien savoir comment vous avez fait pour calculer ou bien pour déterminer les concentrations de chlorure de sodium et de trichlorure, chlorure de sodium, trichlorure d'aluminium. Est-ce que vous avez utilisé des méthodes volumétriques ou les instrumentales? C'est ma première question. La deuxième question, pour la dégradation des raisins en fonction du stress NaCl et trichlorure d'aluminium. J'ai remarqué qu'il y a une différence en ce qui concerne l'anatomie des raisins et comment ça a été infecté par ces deux composés chimiques. Et aussi la troisième question, est-ce qu'on peut expliquer ce phénomène par le phénomène d'osmose? Donc il y a les raisins qui absorbent, bon, il n'y a pas que l'eau, donc l'eau elle est chargée surtout en NaCl. Et on a l'absorption aussi de trichlorure d'aluminium. Je vous remercie.
Donc, je m'excuse parce que les deux dernières questions, je ne l'ai pas entendu très bien. Mais pour la première question, nous avons utilisé le poids molaire simplement de NaCl. Donc, par exemple, pour doser 150 millimoles de NaCl, nous avons dû faire dissoudre à peu près 8,61 grammes de NaCl dans 1 litre de l'eau distillée. Et c'est idem pour les autres concentrations, juste le poids molaire et des règles de 3. Deuxième et troisième question, je m'excuse, mais je ne les ai pas entendues très bien.
D'accord. Je vais recapituler. Vous avez dit qu'il y a la dégradation des résins en fonction du stress de NaCl, trichlorure d'aluminium.
Madame, dégradation de quoi?
La dégradation des résins.
La dégradation des résins.
Oui.
Ok.
On le voit sur la morphologie des résins.
Mais je n'ai pas mis des photos des rhizomes ou bien des morphologies des rhizomes. Nous avons juste calculé le nombre de cellules vivantes. C'est ça.
Ok, d'accord. Et pour l'effet d'osmose, comment vous pouvez expliquer le phonème d'absorption d'aide?
Expliquez quoi, je m'excuse. Comment vous pouvez expliquer quoi?
L'effet d'osmose.
Je n'ai pas bien compris la question.
L'effet d'osmose.
Qu'est-ce qu'il a?
Comment vous pouvez expliquer l'effet d'osmose?
Je n'ai pas parlé d'effet d'osmose, monsieur.
Quand on a des sels minéraux,
Oui.
comme la molécule de NaCl,
Oui.
qu'elle soit absorbée ou là, il faut qu'il y ait un effet d'osmose. Donc, ça passe du milieu hypotonique vers un milieu hypertonique. Donc,
Oui.
le moins concentré vers le milieu le plus concentré. Est-ce que vous avez pris en considération ce phénomène?
Non.
Donc, je ne sais pas, mais il faut voir peut-être ce côté-là. Est-ce
Ouais,
qu'il faut avoir aussi les moyens pour le faire?
Inch'Allah, Inch'Allah.
Inch'Allah. Je vous souhaite une bonne continuité.
Merci, madame.
Any other questions? Okay, Mr. Saadallah, please.
Oui, simplement par rapport à l'effet d'osmose, je pense que la personne qui a posé la question est une chimiste. Chez les plantes, en vérité, il y a un phénomène qui permet la pénétration de l'eau, même si la concentration elle est élevée dans les racines ou elle est élevée alentour des racines, par un compartimentage que la plante juge intéressant du sel dans les espaces intercellulaires et par l'augmentation de la vacuole de la vacuole cellulaire. En vérité, il y a un compartimentage à l'intérieur, ce qui permet en vérité de diminuer ou d'augmenter cette concentration afin de faire introduire de l'eau du milieu extracellulaire. C'est une façon d'adaptation pour la plante pour absorber l'eau, même si la concentration de sel ou des sels, elle est élevée au milieu extérieur. Ça, c'est connu chez les plantes. Merci.
Je vous remercie, M. Saadallah, pour ces explications. Effectivement, moi, je suis une chimiste.
So, thank you, Mr. Saadallah, for this precision. If there is no any question, let me thanks again Mr. Zarrouk for his clear presentation. And the next one is biocontrol of grain pest Tribolium castaneum by the essential oils of Artemisia, Juniperus, and Geranium. Mr. Munir Kharoubi, please.
Mr. Kharoubi, please share your
Yes,
screen.
I'm here.
Okay, because
Yes,
I don't have your presentation.
Really? No, I sent my presentation a few days ago. So and I checked the of Madame Hamid. So my presentation is already there.
Do you have it now?
In my laptop, no, because I'm in the car now.
Okay.
Okay.
Okay. So
Sorry for that. I'm in my car. I am trying to be online to answer if there is some questions.
Okay, so I will prepare your presentation. I will check my account. Okay, so
Yeah, if you didn't find the data, you can see in the laptop of Madame Hamil, because I
no
already
problem,
seen
no
that.
problem. Okay, no problem.
Okay.
So we move to the next presentation, then
With
we
the
will come
I-
back to you. Okay.
Okay.
Okay, no problem. Okay.
Okay.
Okay,
Thank you.
Chemical
Thank you.
conversion of waste biomass into renewable fuel with Sumia Feraza.
Yes, I'm here. Yes, I'm here.
Okay. Thank you.
Yes, I'm here.
The ID.
ID is
Yes.
ID
100.
110.
Good morning, everyone. Thank you very much for being here this morning. It's a big honor to be with you today at this interesting conference. Today, I'm going to give you a short presentation about my work entitled Chemical Conversion of Waste Biomass into Renewable Fuel. Let me start saying a few words about the context of this study. Transportation sector and also waste management activities contribute to greenhouse emissions. Burning fossil fuels and organic waste decomposition in landfill sites release carbon dioxide and other greenhouse gases into the atmosphere. This is causing the Earth's atmosphere to warm, resulting in changes to the climate. Non-renewability and the rapid depletion of fossil fuels coupled with serious environmental degradation lead to surge in new and renewable sources for energy production. Biomass is the best candidate may be used to meet a wide variety of energy needs. The conversion of biomass to such useful forms of energy, solid, liquid, and gas, can be achieved using a number of different processes. All forms of energy generated from biomass contribute to environmental protection. This study focused on biomass generated from olive oil industry. Olive oil industry carries both economic and social importance, especially in Mediterranean countries. Algeria is one of the leading olive oil producers. The extraction of olive oil generates annually huge quantities of byproducts such as solid residues called olive pomace and liquid known as olive meal wastewater. Olive pomace is the candidate biomass for this study. It is a lignocellulosic organic material having high energy content. Olive oil byproducts pose several potential environmental problems in the main olive producing countries. The valorization of these olive residues is the best way to improve the economic status of olive oil producers and to preserve the environment. The main objective of this study is to valorize olive meal pomace by converting its residual oil to biodiesel using chemical process. Energetic valorization of olive pomace aimed to produce renewable energy for transportation sector to replace petrodiesel. Let's now turn to experimental methods. After drying in an oven at 40 degrees Celsius for removal of moisture, and then the dried samples were ground and passed them through a sieve to make them homogeneous. The obtained powder used for extraction of oil by soft-slab extractor for 6 hours using hexane as extraction solvent. The oil was then recovered by distillation and the residual solvent was removed by drying in an oven to obtain a dried olive pomace oil. The extracted oil was then characterized and the result presented in the next table. This table illustrates that olive pomace contains of 15.19% oil and 12.51% moisture. Olive pomace oil extracted has a green dry color. It is an acid oil with a high acidity and viscosity. For this reason, chemical process with 2 steps was used. Let's begin by the first step. Acidification requires the removal of free fatty acids from the olive pomace oil by converting them to ester with methanol to oil molar ratio of 8:1 using an acid. A quantity of oleopomace oil were poured into the conical flask and heated to 60 degrees Celsius for 1 hour under agitation. After, the reaction mixture was allowed to settle. The treated oil and acid methanol phases were separated. The second step is used to transpecify treated oil at a molar ratio of oil methanol 6:1 and catalyst concentration of 1% vapor weight for 80 minutes at 60 degrees Celsius under agitation. The reaction product mixture was allowed to stand for many hours to separate into the biodiesel phase and glycerol. Warm water was added to the collected biodiesel, washing to remove impurities. The washing was repeated until no trace of soap found, and final product dried in an open beaker to obtain pure biodiesel. The obtained cracked pomace oil and its biodiesel was characterized by chemical and physical standard methods. The obtained result shows that the used chemical process with two-step gives an ester yield of 97.2% and reduce both of acid number and viscosity. The other properties of produced biodiesel fulfills the international standard. The infrared spectra of both oleopomace oil and its biodiesel were almost similar, but various differences could be observed for identification purposes. The shifting of absorption peaks of the oil sample at 1743 centimeters -1 is significantly more intense in biodiesel than in oleopomace oil. The disappearance of peaks at 1710 and 1162 centimeters -1 from the spectrum of oil and appearance of new bands in the biodiesel at 1437, 1195, and 1168 centimeters -1 indicate the conversion of oil into biodiesel. As a conclusion, in this study, oil extracted from oleopomace was tested for biofuel production by chemical process through two steps. The results indicate that oleopomace oil could be used as a good source of renewable energy for transportation sector. The results of biodiesel characterization shows that the fuel fulfills most of the international standards, so can be used as a possible candidate for replacement of petroleum diesel. Thank you so much for your kind attention.
Thank you, madam. Any question?
J'aimerais bien poser, vous avez montré dans le domaine de la chimie, j'aimerais bien poser quelques questions. Je vois que vous avez utilisé les déchets de l'huile d'olive donc pour construire un fuel qui remplacerait le pétrole. C'est un travail très intéressant pour conserver l'environnement.
Moi, j'ai rien. J'entends pas.
Elle vous entend pas.
Oui.
Okay. Just a moment, please.
Désactive ton micro.
J'ai activé.
Et maintenant, est-ce que vous m'entendez? Est-ce que comme ça vous m'entendez?
Oui, madame.
Je vois que vous avez utilisé les déchets de l'huile d'olive, déchets qu'on extrait de l'huile d'olive, qu'on récupère après l'essorage de l'huile d'olive, pour déterminer un fuel qui remplacerait le pétrole.
Madame, votre question, c'est pourquoi? J'ai pas. C'est pourquoi?
Je n'ai pas encore posé la question, pas encore.
Oui, oui, oui. Puisque madame مانيش نسمع très bien معلش. Oui,
وهكذاك تسمعي.
oui, oui, madame.
Très bien. Donc
Très
là, je vois que vous avez passé par plusieurs étapes. Vous avez fait passer votre échantillon par plusieurs étapes.
Oui,
Et vous avez fait l'analyse par infrarouge pour déterminer le groupement fonctionnel. Par exemple, le C de liaison O, OCH3. Mais
et surtout, madame, la fonction ester. Surtout la
Très
fonction ester, oui.
est-ce que vous n'avez pas essayé d'analyser vos échantillons après chaque étape? Je vois que vous avez fait plusieurs étapes.
Oui, madame. Donc, oui. Madame, toujours l'analyse. Donc, qu'est-ce que j'ai fait? L'extraction, donc, j'ai fait la caractérisation de l'huile d'olive. Et après, la conversion en biodiesel. Donc, j'ai fait, madame, comment, l'infrarouge, l'huile d'olive, l'huile de graineaux d'olive. Alors, en parallèle, la biodiesel. J'ai fait deux analyses, l'huile et son ester.
Non, vous ne m'avez pas compris. Vous avez,
Oui.
pour l'estérification, vous avez passé par plusieurs étapes. À chaque fois, vous avez eu un échantillon.
Oui, madame. Donc, oui, je comprends votre question. Madame, les conditions, les خدمت بيهم pour l'estérification et la transestérification, ce sont des paramètres déjà optimisés. Avec des travaux récents. Donc, j'ai utilisé seulement les conditions optimales ou directement خدمت بيهم. C'est-à-dire, ماشي أنا اللي درت l'optimisation. هما داروا هذيك l'étape, madame. Chaque étape, يديروا une analyse pour avoir هذوك les conditions.
Très bien. Et je vois que vous avez comparé avec la courbe ASTM. La courbe ASTM, elle est la plupart du temps, elle est utilisée pour caractériser un pétrole brut. Et vous l'avez comparé à cette...
J'ai comparé seulement l'huile avec son ester. Est-ce qu'il y a une conversion ou non? Voilà. Madame, qu'il y a les acides gras libres, etc. Donc, tout ça, après la transesterification, tout ça, il y a une disparition de tous ces pics ou des transferts le biodiesel, l'ester de l'ester, la fonction ester.
Ok, merci.
Voilà, est plus intense, madame, est plus intense. L'ester, plus que lui.
Merci pour ce travail, c'est très intéressant.
Merci, madame. Merci.
Thank you. Other questions? Other questions? Thank you, Madam Firanza, for your presentation.
Thank you very much. Thanks.
Thank you again. The next one is with the ID of 134, bioadsorption behavior of methylene blue onto Rosmarinium officinalis, process optimization, kinetics, and equilibrium. This is Taous Hamad. Please.
Taous
Hamad.
Taous, are you here, please?
Okay, then the presentation with the ID 145, Mrs. Bouhallal Sara, determination of pearl millet starch functional properties isolated by conventional wet milling and high intensity ultrasound assisted isolation.
I think she organized another room. Shall we pass to the
which one? 154?
Yes.
Effects of calcium chloride on adjumalicin projection and biomass accumulation in Catharanthus roseus transgenic hairy roots. بنيامي رقية.
Dr. Binyami.
Good morning, everyone.
Good morning. Okay, thank you. So I will share your presentation.
Good morning, everyone, and welcome to my presentation. I am Rukia Bini. I am lecturer at UNESCOBA as I am senior researcher at Algeria. In my presentation, I would like to report on effects of calcium chloride on advanced production and biomass accumulation in catalytic transgenic hair roots. Catalytic is still one of the most important sources of terpenoid under alkaloids, including anti-cancer molecules as vincristine and vinplastin, and anti-hypertensive molecules as adjuvants. The layers of these alkaloids, their high cost presents a serious challenge for biotechnologists to find a solution for improved yields and reduced production costs. High-root scripture represents a really alternative to improve the performance and reduce the unit cost of the production of these valuable alkaloids. In our work, we have chosen high-root cultures for many advantages. A phenotypic variation, in vitro development in a fetal hormone-free environment, and the genetic and biochemical stability. For these reasons, high-root cultures are considered an attractive system that could play a potential role in the pharmaceutical industry to produce active biomolecules. Several strategies are proposed to optimize the production of biomass and secondary metabolites. Among these strategies, we find elicitation of cell and tissue cultures, treatment with an expressor regulator, and overexpression of genes responsible for the biosynthesis of the metabolite. In this work, we are interested in elicitation with abiotic agents such as calcium chloride. The objective of this work was to determine the effect of calcium chloride as the abiotic elicitor on biomass accumulation and adjuvant production in high-root cultures. Firstly, we started by establishment of high-root cultures by infection of experts with agrobacterium resistance, and selection of high-root line which represents better growth. The transgenic nature of selected high-root line was confirmed by detecting the presence of a ROLB gene by PCR amplification using the specific primers. In order to study effect of the elicitation on accumulation of biomass and adjuvant, two baseline of hair roots showing good growth and on liquid medium were selected to apply calcium chloride as abiotic elicitor. The elicitation was applied during the exponential phase of growth on liquid culture. After an elicitation times of 24 hours and 48 hours, the effect of the elicitor is evaluated on the basis of the dry weight of the plant material and their adjuvant content. The identification and determination of adjuvant in mechanical X-ray of hair roots is carried out by liquid chromatography coupled to tandem mass spectrometry (ICEBB2021). This step is performed in laboratory LBU at University of Lorraine in Nancy, France.
cultured for 3 days with agrobacterial residences have the best abilities to induce herons. This efficiency of transformation by the agrobacterium can be explained by hypervirulence of agrobacterial strains and the presence of meristematic cells in the bud, which can promote rapid transformation. Two lines, L54 and LP10, were selected on the basis of their high biomass production. PCR verification of the transgenic nature of the induced shows the presence of a ROB gene that in the two selected root lines. Therefore, we can consider that all the root lines that express this gene are transgenic. The bud selected high-root line showed almost the same biomass accumulation profile through the culture cycle. The exponential rise with a large-scale biomass accumulation was ranging from the 23 to 30 but the LP10 line showed a quick and high growth in which the dry white reached 0.35 grams, threefold than initial white. On the other hand, our results showed that the contents of admalisine are significantly higher for LP10, with a content of 430 milligrams per 100 grams of dry white, compared to L54. In addition, the maximum production of admalisine coincided with a late exponential biomass growth phase. A result obtained after elicitation of two selected root lines, IP10 and L54, with the salt calcium chloride shows slight increases in dry white of herons after elicitation with 3 millimole of calcium chloride for 24 hours. Concerning the effect of calcium chloride on the content of the admalisine in heron, the highest content is obtained with LP10 line elicited with 3 millimole of calcium chloride for 48 hours. On the other hand, the best change in admalisine content is recorded by L54 line after treatment with the same concentration, 3 millimole of calcium chloride for 48 hours, which improvement is 39% compared to control. Our results obtained from two selected high-root lines suggest that the evolution of accumulation of admalisine is positively correlated with development of biomass growth. Moreover, our study showed that LP10 line is the most effective and promising line to continue optimizing admalisine production. Our results from the elicitation of herons show that LP10 line gave the best levels of admalisine due to their level already very high. However, line respond best to elicitation, have the best response to elicitation is L54, which the best improvements are recorded with this line. Thank you for your attention.
Thank you, Madam Binyami Rafaya. Any questions? Okay, thank you again. The next presentation is about etiological and epidemiological study of food disease of the citrus in Algeria. Ahmed Faiza.
Ahmed, please raise your hand if you are present. Ahmed Faiza, no.
Then the ID number 114. Madame Kerbel Safia.
The ID 114 are, yes, she is here. Good.
Okay.
ما عندي أنا bon. كيفاش la narration? Please, Kerbel. Kerbel? You send us the PowerPoint without narration. So you have to speak now. Give us the talk. I will present your. I will share your presentation, and you will talk. Okay? So you have to enable your microphone. Kerbel. I will try to fix the problem. Okay.
So maybe we can go to the last one. The ID 178. Madame Zouari Nafila.
To present this work entitled Evaluation of the Insecticidal Activity of the Essential Oil of Cinnamon Cassia against Colossobruchus maculatus, realized by Nafila Zouari, Munir Kharoubi, Soufiane Chouch, and presented by Nafila Zouari. Storage food can be attacked by insects, fungi, and rodents. The insect's damage is the most significant. The Colossobruchus maculatus is the major and the most serious pest of legumes. However, the management of insect pests in many storage systems relies principally on applying synthetic insecticides. Use of a synthetic insecticide is currently the most effective way to prevent the infestation of stored product pests. However, continuous and heavy use of these chemicals has caused adverse effects on non-target organisms and the development of pesticide resistance in some stored product pests. To solve this problem, many researchers have discovered alternative pests and chemicals management projects derived from plants. Several plant projects have been investigated for insecticidal activities. Fumigation of some essential oils and individual larval development at different stages of growth. In the present investigation, essential oils of cinnamon cassia was tested as a contact insecticide from insect pest Colossobruchus maculatus in laboratory conditions. The objective of our work is the first one to determine the major components of the essential oil of cinnamon cassia by CGNS analysis. The second one to test the insecticidal effect of this essential oil against Colossobruchus maculatus. question that rather does the essential oil have insecticide activity, and does the concentration of essential oil and does the percentage of insect mortality. The essential oil is a mixture of organic compounds represented by monoterpene, sesquiterpene, terpene, and other compounds such as ester and aldehyde. It is obtained from natural material by extraction in several ways. It is characterized by its small partial weight and differs from mixed oil by its volatility. It is found in different parts of aromatic plants. There are several methods of extraction. The most commonly used being distillation with hydrodistillation and steam distillation, using the dioxide carbon and microwave. Cinnamon is a high plant consisting of stems, leaves, and flowers. Trilate in Latin, Cinnamon zianica. Is synonymous with Cinnamon verum and Cinnamon aromatica. Shown in the photo, the cinnamon tree has a strong trunk and a reddish-brown bark. Its shape is round and dense. The leaves are evergreen, aromatic, and shiny on this riverside. In the experiment, we used adults of maculatus from a mass breeding carried out in the oven 35 Celsius. The mass breeding of maculatus was achieved in glasses containing unchiquita grains. The glasses were maintained in the dark in a laboratory oven set at 27 Celsius and 65 of relative humidity. The essential oils are obtained by hydrodistillation. As shown in the figure, the yield of essential oil increases with time. For an extraction time of 3 hours, the yield is 2.3%. Whereas for 1 hour 30 minutes, it is reduced by 7-8%. The oil is essentially composed by trans-cinnamaldehyde, cinnamyl acetate, and methoxynamaldehyde. The toxicity result of the extracts in cinnamon at different concentration against agent maculatus are represented in the table. Important efficiency after 36 hours in accordance with concentration 0.13 gram per milliliter and the dose 0.013 gram per milliliter with a mortality average of 90% and 40% respectively. For the concentration 0.13 gram per milliliter caused 5-7% of mortality after 6 hours, 5-3% after 4 hours, and 30% after 2 hours. This study was conducted on insecticidal effect of cinnamon cassia essential oil on the legume insect Colossobulgus maculatus. The results show that at a concentration of 0.13 gram per milliliter of essential oil, insects reach a mortality rate of 57% after 6 hours and 93% after 36 hours. Our study makes it possible to verify the insecticide effect of natural substance in order to avoid the use of chemicals in food source. Thank you for your attention.
Thank you, Madam. Next question. Mr. Sadullah.
Rebonjour. Donc, c'est un très bon travail d'investir des insecticides qui sont un peu naturels par rapport à ce chimique. Bon, j'ai compris que l'huile essentielle utilisée est une huile essentielle à partir de la plante que l'on a citée. Je voudrais savoir pourquoi vous utilisez une huile essentielle. Est-ce qu'elle n'est pas absorbée par les graines? Est-ce que vous avez testé un peu ceci sur les graines? Est-ce que ça ne va pas changer? On sait très bien que cette plante présente un arôme très fort. Est-ce que ça ne va pas changer? Est-ce qu'il n'y a pas une alternative d'utiliser sous forme de poudre ou sous forme de gaz pour éviter qu'il y ait une absorption par les graines? Parce que c'est important pour la nutrition et pour l'alimentation. Merci.
Merci, M. Sadallah, pour votre question. Nous avons fait un essai au niveau du laboratoire en utilisant l'huile essentielle. Mais si vous voulez, nos ancêtres et nos grands-mères, pour conserver les graines secs, qu'est-ce qu'elles faisaient? Elles utilisaient soit le sel, soit le poivre noir. Donc là, c'est vrai, si on met des gouttelettes d'huile essentielle, c'est vrai que ça va influencer sur l'odeur et la qualité dégustative des graines. Mais on peut bien les remplacer par les brins de cannelle à l'intérieur du bac où on conserve nos grains secs. D'ailleurs, on peut aussi, si on a un magasin pour emmagasiner, on peut évaporiser l'huile essentielle de la cannelle ou bien d'autres plantes pour conserver ces grains secs.
Oui, bon, de mettre les cannelles sous forme de bâtonnets, c'est bien. De les mettre également sur poudre, généralement, les gens, quand ils achètent, même si c'est des légumes secs, etc., ils vont les laver. Mais est-ce qu'il ne peut pas voir, il faut pas voir si on ne peut pas l'utiliser sous forme de gaz, par exemple, parce que ce qui est important, c'est que généralement, ces aliments sont stockés dans soit des silos, soit dans des grands hangars. Et pour éviter vraiment qu'il y ait un contact direct ou qu'il y ait un changement gastronomique ou des légumes secs, il est préférable quand même de penser, et je pense que ça peut être une perspective pour vos recherches, d'essayer de voir si c'est pas c'est pas possible d'utiliser sous forme de gaz carrément, une fumigation d'une gaz.
Oui, justement, c'est possible. D'ailleurs, nous avons un matériel pour évaporiser les huiles essentielles. On les mélange avec de l'eau, on bouillit l'eau, il y a des vapeurs, et ces vapeurs peuvent stériliser l'ensemble ou l'environnement qui entoure ces grains secs. C'est possible de faire.
Merci, madame.
Je vous en prie, je vous remercie aussi.
Other questions? So if not, we thank again Madam Zouari. And we go for the next presentation with the ID 88, Mr. Kharoubi Munir.
Yes, sir.
Algeria. So they implement to build the work and set up a control repairs to 100% by the certain modes of repairs with 100%
I think the quality of the sound is
Very good.
yes, it's so bad. So I will ask again, Mr. Kharoubi, if you can share your screen.
I didn't yet arrive to my home, so I can read when the video it starts. So it's okay.
You can read it?
Yeah, I can. I try to read
Okay.
on my phone. Yeah. Okay.
So please
So,
proceed.
okay. Morning, everybody here. I'm Karobi Munir, a member at Ecobiology Animals Laboratory at the Training School of Teacher in the Cuba. And I am pleased to present my work under the title Biocontrol of Grain-Based Tribolium castano by the essential oils of three plants. It's Algerian plants, Artemisia, Junipers, and Geranium. Okay. So, as an introduction, the botanicals, botanicals are basically secondary metabolites that serve as a defense mechanism of plants to withstand the continuous section pressure from herbivore predators and other environmental factors. So plants produce essential oils as terpenoid, alkaloid, and another, which many have medicals, insecticides, and larvicidal activities. Plant essential oils in general have been recognized as an important nature resource for insecticides. So in recent years, and due to the lack of novel insecticide and the high cost of synthetic insecticide, the use of many synthetic insecticide formerly used in the pest control program has been limited. Therefore, the search for alternative to synthetic chemical insecticide and nature extract of and essential oils for vector and pest management that pose a little threat of or to human and environmental health has increased. So many research have thought that plant parts, essential oils extract or powder mixed with grains reduce insect oviposition, eggs hatchability, and post-embryonic and development inability reproduction and induced mortality of insect eggs and the progeny production of stored product insects. So the aim of this presentation studies was to determine the chemical composition of these three plants and try to evaluate the importance of this molecule and the compound toxicity against an important pest of stored grain tribolium cassa. So as a material and methods, the red flow betas, adult and the larva were weighed with flow and the laboratory condition of temperature and humidity. Okay, about the next step, please. About the extraction method of plant extraction of essential oils. Freshly collected samples of aerian parts, stem and larvae and leaves, of Artemisia, Juniperus and Geranium were cut into a small piece using acetone and hydrodistillated for 3 hours using a Clevenger type machine. Also, the collect essential oils are extracted by some molecules of sulfate molecule, yeah. And about the GCMS analysis, the constituent of Artemisia, Juniperus, and Geranium essential oils were analyzed by gas chromatic mass spectrometry in laboratory in French with some friend. Yeah. And about the assessment of toxicity by contact effect, the insecticidal effect of essential oils extracted against the first instar of larvae of Tribolium castinum was evaluated by means of contact toxicity, and a range of volumic doses were prepared at 10%, 20%, 30%, and 40% for each extract. Okay, so each filter paper was then placed inside a glass petri dish with 20 larvae of Tribolium castinum at optimal temperature condition. As a result, we're trying to resume our result and talking about the little doses of essential oils test against Tribolium castinum larvae. So this result showed two of these plant extracted tests, Artemisia and uranium, were testing toxicity. This present the weakest concentration of little doses at 0.003% and 0.006 respectively. On the other hand, the extract of juniperus was the less toxic with a value of 0.09, pardon. So our research showed the insecticide effect showed a remarkable insecticidal effect and characterization of Artemisia and Juniperum against the Tribolium castinum. About the chemical analysis of GCMS, the Artemisia juniperus showed a major composition of chrysanthemum at 31.40%, followed by alpha thion and camphor, then the limonene with 5%. Okay. So about the juniperus results of GCMS analysis showed an identification of 12 compounds. The major compounds were alpha benene at 64.18%, and the second compound presented by the myrcene with 5%. And in the second table, it's about the geranium GCMS analysis. Showed. Do you hear me?
Yes.
Okay. So, but I'm waiting. Yeah. Okay. About the second table, it's about the chemical composition of geranium essential oils. It showed about 15 compounds were identified. The major compound presented by the citrinol at 36%, then the geraniol at 16.63%, and followed by the isominton at 8.82%, and the citrinol, the citronellol format at 7%. As a conclusion of this investigation, it affirms that the essential oils of Artemisia and geranium can ensure the protection against Tribolium castinum. Consequently, they can be exploited as a bioinsecticide agent. About this study, therefore opens up a wide range of possibility for assessing the insecticidal and toxic effect of the essential oils studied on adult Tribolium castinum. Additional experiments are needed to clarify the nature of the compound responsible for the activity to optimize activity doses. At the end of this work, we offer some reflection and recommendation under the form of the prospect for good exploitation of the byproducts of these spices to preserve. Also, establish the chemical composition of this studied essential oil. And thank you for your attention.
Thank you, Mr. Kharoudi.
You're welcome.
So please, if there is any question. Okay, so now presentation concern Madame Kerbel Safia, ID 114.
Bonjour. Bonjour.
Okay, you can start, please.
S'il vous plaît, vous avez partagé l'écran. C'est bon, vous avez partagé la diapo. C'est pas la peine de la partager d'ici.
C'est pas la peine, on la partage nous-mêmes.
Allez-y, madame.
S'il vous plaît, c'est pour faire passer les diapos. C'est comment?
Je vais les passer moi-même.
C'est une vidéo.
C'est pas une vidéo. OK?
D'accord. Donc, I have the honor to present to you my work entitled Comparative Study of Biocidal Activity of Olive Oils extracted from different regions of the Kabylia on adults of Cytophilus oryzae under controlled laboratory conditions. Donc, introduction. S'il vous plaît, est-ce que vous pouvez passer? Donc, among the pests of wheat greens during storage, Cytophilus oryzae is a serious pest of stored green worldwide due to the quantitative and qualitative losses that it causes. The infestation of stocks by Cytophilus oryzae causes weight loss, a decrease in essential amino acids, a decrease in the germination capacity of greens used, acid, and a reduction in plant vigor at emergence. Control measures are currently based on the application of chemicals because of their effectiveness and low cost. The use of conventional insecticide has caused adverse effects on the agroecosystem, such as the development of insect resistance, resurgence of secondary pests, and environmental contamination. Among vegetable oils, olive oil, due to its high oleic acid content, detected as an insecticidal component, could be used as a biopesticide in insect pest management. For that, the aim of this work is to study the fatty acid composition and the insecticidal activity of oils according to origin of plantation against one of the main insect pests of stored green, who is Cytophilus oryzae. The next diapo, please. Donc, the extra virgin olive oils used in this work come from Shumlal, the main Algerian olive variety at the same stage of maturity in the 2016 and the 2017 season. 4 representative regions of olive oil production in Algeria were selected to obtain the extra virgin olive oil samples. Donc, it's about Tadmeg, Madqa, Mshdala et Beshloul. Donc, olive oil samples were obtained using a laboratory scale oil meal consisting of 3 basic elements. donc a hammer crusher, a thermometer, and a pulp centrifuge. The insect, donc the next diapo, please. Donc, the insect, we have used adults of Cytophilus oryzae belonging to the genus of Cytophilus and the species of Cytophilus oryzae. Substrate used is seeds of softwheat. For bioassays, donc each olive oil extracted was mixed with soft wheat grains in petri dishes, with the 3 concentration, 3 doses, c'est 0.1, 0.2, and 0.5 milliliter. Then 20 insecticide adults were introduced into each dish, which was immediately closed. Mortality was assessed after 1, 2, 3, and 4 days after treatment application. On day 45, samples of treated control grains were taken to evaluate weight loss and germination capacity. Donc, for results, donc for results of fatty acid composition of the 4 oils tested, the major fatty acids present in Schindler olive oil were oleic acid, linoleic, palmitic acids. Oleic acid is the main monounsaturated fatty acid, and it is present in high concentration. Palmitic acid content, the major saturated fatty acid in olive oil, varied between 15 and 17 and 18% according to the plantation region. Concerning linoleic acids, the highest percentage was observed in extra virgin olive 2, whereas the lowest was found in extra virgin olive oil 1. Concerning the contact toxicity of olive oils against Cytophilus oryzae, donc in contact mortality assays, donc after 24, 48, 72, and 96 hours, the ascending of concentration of the 4 oils caused increased mortality of the beetles. After 45 hours of exposure, all oils at the highest dose are very toxic to Cytophilus oryzae. The mortality are ranging from 72 to 95%. Donc, concerning the second parameter, c'est progeny emergence. Progeny emergence. Donc, the highest number of emergence are observed in the control lots, with an average number of 92 adults. Moreover, the number of progeny production is inversely proportional to the dose used. In fact, this number decreases as the dose is increased and becomes 0 at the highest dose with all treatments. Concerning the third parameter, c'est grain weight loss. Donc, according to the results obtained, we observed that Cytophilus oryzae caused high weight losses in the control lots. On the other hand, in lots treated with oils from different regions, a considerable reduction in grain weight loss rates is observed as the treatment dose increases. For all oils tested, treatments with the highest dose completely preserves the soft wheat seeds from pest attack. Concerning donc the last parameter, seed germination, donc the tests carried out show that the germination capacity of wheat seeds from untreated and infested control lots reaches 92%, while this rate decreases as the dose increases in seeds treated with the various oils ranging from 79 to 45%. For conclusion, for conclusion, donc we can say that the four oils tested show, donc the result shows that the four oils tested are highly toxic to Cytophilus oryzae. In fact, this toxicity is increased as the dose increases. The study confirmed the agro-phytosanitary potential of Algerian olive oil, which can be used locally as a biopesticide for the protection of wheat seeds, whose preservation is a major challenge for Algeria. Since Algeria has a diversified olive growing heritage, the valorization of these plant extracts as part of an integrated control program in the eco-chemical fight against insect pests of stored grains in order to reduce the significant economic loss recorded each year in stored warehouses in an interesting option. And thanks for attention.
Thank you.
Okay. Do you have any question? Or comments? You can use the chat to write if you want a question or comment to Dr. Kerber. Okay, thank you so much, Sophia Kerber, for the presentation.
Merci.
So,
Ahmed Faiza.
Ahmed Faiza. No, she isn't here. Samir Hajmiloud. No, we don't have Hajmiloud here. He just attend the session. Okay, Selma Baka. Open your microphone. Okay, Selma Baka.
Good morning.
Good morning.
Bonjour, madame. Moi, c'est
Bonjour.
Hajmiloud. C'est Hajmiloud.
Okay, wait a moment, Mr. Samir.
Okay. Thank you.
Yes.
Je peux présenter, s'il vous plaît?
Oui.
Merci.
J'ai pas la présentation, votre présentation, parce que apparemment elle a raté la session de la matinée, c'est ça?
Non, moi, je suis programmée pour la session d'après-midi, mais je n'ai pas pu rentrer dans la Rooms 5.
Room 5, c'est pour la matinée, non pas l'après-midi. Hein.
Si mentionné, madame, c'est 1h, 13h jusqu'à 16h.
Donc, c'est pas room 5, c'est room...
Room 1.
Which thematic, please?
Room 5, madam. Room 5.
I ask you, which thematic? Which track?
Climate change, adaptation in water, soil.
Track 4 or 5?
5.
Track 5? Okay. Okay, not now. The track begins at 1:30.
Okay, it's okay.
Okay, thank you.
S'il vous plaît, madame.
Yes.
Toujours rooms 5.
Yes.
D'accord, ok, madame. Merci
just,
beaucoup.
Please, you send us your presentation.
Je vous ai envoyé la présentation, madame.
I didn't find it. Sorry. Okay, I will check later. Okay.
Je vais la renvoyer. Ok, merci ça.
No problem.
Madame, c'est Hajmiloud. Si vous voulez, je peux faire la présentation. Bon, mais j'ai le même problème. C'est-à-dire que je suis programmé l'après-midi. Hein. Track 5.
Track 5, c'est cet après-midi normalement.
Oui, parce que vous m'avez appelé maintenant. Parce que vous m'avez appelé, donc...
Ah, d'accord. Non, je pensais que vous allez participer. C'est tout.
C'est quand vous voulez. Donc, j'attends jusqu'à l'après-midi. Donc, je suis le programme.
Oui, exactement. Merci beaucoup, Monsieur Hajmi.
Okay.
So, I don't know if you have any comments, No? Okay, so thank you for your participation. Thank you for your attendance. and hope you all the best, and hope you can publish all the results, inshallah. Thank you to Mr. Dr. Buhenna from CLAPSE, and have a nice day. Thank you. We wait your feedback, so please send us an email on ICEBB or email Malika Adidi. Okay? Okay, thank you. Thank you so much.